Today we're going to be going over section 4, classification of neurons and the neuroglial cells. Neurons we've kind of already talked about, but we're going to look at today specifically the structural differences. There are three types, as you can see on the screen, that we're going to cover. We're going to look at the functional differences of these neurons and what they do. And then we're going to look at how do we classify the neuroglia, or the support cells to the neurons. And we're going to look at the peripheral nervous system, the central nervous system, and then finally kind of wrap up with how do cells um, regenerate or make new cells. So let's take a look at this slide here. Certainly you can see the three different types of cells. You're most familiar with the multi polar cell, which is right here. This one you learned about as a sophomore. And so today we're going to look at the other two cells, the unipolar and the bipolar cells, which are over here. So we'll start with the new one um, as we take a look at that. But here's kind of a listing of those three cells, the bipolar, unipolar, and multipolar. They vary in size and function. So what do they do? And so we're going to look at those differences between these three neuron cell types. The first one we're going to talk about is the bipolar neuron. Note there are two extensions. There's one extension here and the second extension here, and the soma, or the cell body, sits right here in the middle. So the signal comes all the way across. And so on one end, we have the dendrites. Remember, dendrites always carry information to the soma. So whether you're sensing light, sound, pressure, an odor, whatever the case may be, the dendrites are going to have some sort of sensory receptor, whether it's a chemoreceptor picking up a chemical, a light receptor, a rod or a cone sensing some sort of light or color, or a sound receptor, whatever the case may be. That's going to be on the end of this. We're going to generate an action potential, and we're going to send that electric signal to the soma in this direction. Then that signal is going to pass through the soma and go through the axon, where it's received in the central nervous system. And we do something with that. Whether that's the brain, typically that's the brain, at least in the case of our eyes, our nose, and our ears. But remember, the central nervous system has to do with the brain and the spinal cord. So this is a bipolar neuron, typically involved with sensory type function. Next, we have a unipolar neuron. This is where the soma sits off to the side. So it's not in line. It's not as big as the one on the multipolar. So that soma sort of sits off to the side. And then we have two processes, really one process, but two branches, I should say. The peripheral process, that's more on the periphery, more distal. And so we're going to receive some information on this part of our neuron. And so that's where the dendrite is. Again, we're going to sense something, some sort of sensory type situation, and that information is going to be carried along the axon in this manner. And then we have the central process, which is going to further carry that information on to the central nervous system. So here would be the axon terminal end ending up in the brain or spinal cord, and again the dendrites here. So the peripheral process sits more distally, the central process sits more proximally, depending on the situation. One thing that's unique about the soma, or the cell body, which we looked at right here a little bit earlier, typically they sit in things called ganglia. And these ganglia sit outside your brainal spinal cord in areas throughout your body. And ganglia are just groupings of these somas in certain areas. So we get a whole bunch of neurons and a grouping of them, and that's how we create a nerve. Remember, a neuron is a single cell, a nerve is a bunch of cells, and a ganglia is a bunch of those somas of those cells. So that would be the unipolar neuron. Next, the one you're probably most familiar with from your sophomore year, the multipolar neuron. Here you can see that one. There's many extensions, or many dendrites, that we've talked about already. And these dendrites lead toward the cell body. And then there's one axon that's going to lead away from the neuron. These are the most common neurons in our brain and spinal cord. They make up 99% of the neurons. Not to be confused with our neuroglia cell, but of our neurons, most of them are multipolar neurons. And they're found mainly in our brain and our spinal cord. Um, so there's one axon that carries information away from the soma. There is only one axon, but we can at times have multiple collaterals. And so these are just branches. And um, these branches that come off make connections with other multipolar neurons, and that's how you're able to create memories or embed things better or work around connections or um, create when you assimilate new information. And so we can have collaterals branch off, be created off these neurons um, to make better connections with things. And so that's the multipolar neuron. So as we take a look at schematic here, how do things work, and we're going to break down how do these neurons function. Certainly you can see we've talked about already, the nervous system is divided into the central nervous system. Remember, that's the brain and the spinal cord. And then we have the peripheral nervous system. These are nerves that are either coming to the brain or spinal cord or leaving the brain or spinal cord. And so we'll break down this schematic a little bit. But out here we're sensing something. Maybe you step on a tack on the floor with your foot. You sense that pain, and our neuron picks up that information through a sensory receptor. The dendrites send it, and our axon carries it to the brain or spinal cord. Okay, we're receiving some effect from the thumbtack on the floor and stepping on it here. And so these are called afferent neurons, sensory neurons. They're carrying information to the brain or spinal cord. Okay, and so we're sensing that coming in our brain and spinal cord. We're going to do something with that information. We'll send it up to the brain or spinal cord, or we may even, in fact, generate a reflex response, which we'll talk about later. And then we're going to do something about that. So now we have a neuron causing some effect. And as you take a look over here, we're going to send a signal back, causing an effect to a muscle or a gland. Maybe it's your stomach, maybe it's your lungs, maybe it's your heart, maybe it's your adrenal gland. In this case, after stepping on the tack, sensing it, we send that information to the spinal cord. We generate a reflex activity. It comes down the spinal cord, and then we have a motor neuron leaving the spinal cord going to your leg muscles, causing you to pick your foot up off that tack. So that's kind of the pathway. And so we're going to look at the functional differences between these three types of neurons: the sensory neuron, the interneuron, and the motor neuron. So in this next slide, I believe we have the sensory neurons. Their function is to sense things. They're found in the peripheral nervous system. As noted before, they're afferent. Some of them are bipolar, but we also have some unipolar, as we've kind of seen here. They carry sensory information either from a chemoreceptor, pressure receptor, thermal receptor, mechanoreceptor, all that, and they're going to carry information and put it into our central nervous system, and there we're going to do something with it. Most of these are in our sense organs: our eyes, our ears, our nose, our tongue, etc., etc. Also our skin. Chemoreceptors, you get into some poison ivy or something and you have a chemical reaction. Thermal receptors, the sun comes out and you warm up. Proprioceptors, where are things at in space and time? So all those things, as you can see here on the bottom, we're sensing some activity and we're carrying that information to the brain or spinal cord with this afferent sensory neuron. Some effect is taking place out here. Next one is an interneuron. Interneurons are found in the central nervous system, so either in the brain or the spinal cord, and they link other neurons together. Okay, typically they're multipolar, as you can see here, and so they're going to link sensory neurons to other interneurons, interneurons to other interneurons, and these are the things that make up the most of your cerebral cortex. And so what these do is allow you to make connections and make associations. Sometimes they're called internunential neurons. But they're going to help you assimilate and what is your response. Um, these are responsible for making all kinds of connections. So maybe somebody says the word cat, and then you, with your interneurons, make all sorts of um, connections based on just that single word cat. You might think of dissecting the cat earlier this year. You might have a specific type of house cat. Maybe you live on a farm and have all kinds of cats on the farm. You could have maybe you're in charge of cleaning the litter box. Maybe think of tigers or cheetahs, other things in the cat family. All those things come into play, and all these interneurons connect, relating all those experiences that you've had with a cat, seeing a cat, the color of a cat, the sound of a cat, relating all those experiences. And that's what the interneuron does in making all those sorts of connections. The last one is the motor neuron. The motor neuron typically is a multipolar neuron found in the peripheral nervous system. Actually starts in the central nervous system here and goes to the peripheral nervous system where it carries out, whoops, that's a P, where it carries out some effect, causes your muscle to move or causes a gland to perform or an organ to perform. So you're going to make a hormone, you're going to make a neurotransmitter, you're going to perform some activity, whatever the case may be. So you're going to carry out some sort of effect. And so this is output information from the effector to a muscle or gland, carrying information away from the brain or spinal cord. So here's the diagram again, looking at all those sensory information, carrying information to the central nervous system, interneurons doing something with that information, and then lastly, motor neurons carrying information, causing some sort of effect. So sensory, integration, effect. Next, we're going to talk about the neuroglial cells or support cells of the nervous system that lend some support. You can think of neuroglia cells as sort of the glue that holds things together, the support network, the framework that keeps neurons in place and does another associative type functions. So we'll talk about those. There's 4 different cells in the central nervous system and 2 different cells in the peripheral nervous system that we're going to look at. So the peripheral nervous system has the Schwann cells. We've already talked about Schwann cells in section number 3. Schwann cells are the cell that wraps around an axon and produces myelin, myelinating those cells, those axons of the cells in the peripheral nervous system. One thing to note here, one cell can only operate around one axon. That's one thing you need to note. Whoops, I don't want that. Try not to make a complete O there. That didn't work. One axon in the peripheral nervous system. You'll find that's different when we talk about the oligodendrocyte in the central nervous system. The oligodendrocyte does a similar function, but can wrap around multiple axons. And then we have satellite cells. Satellite cells surround and support the cell bodies, the somas, of the neurons in the peripheral nervous system. And they do this in the ganglia, groupings where the cell bodies is, support them, hold them in place, and we have large clusters of them. So that's what the satellite cells do. The central nervous system, there are 4 types of cells that we're going to talk about that make up the majority of the brain and spinal cord. The first of which is called an astrocyte. Did I not have, I was going to try and make this a little bit bigger, but I guess, there we go. As you take a look at the astrocyte, astro means star-shaped. You can see that. I'll slide this over just a little bit right here. Here's an astrocyte right here. It has kind of a star shape. It connects to this blood vessel coming right through right here and to multipolar neuron right over here. So it's going to connect those two, form a barrier between those two, and we'll talk about um, the further functions of those in just a second here when we get back. So what do the astrocytes actually do? They nourish the neuron. They help get nutrients to the neuron from the capillaries to the neurons. They give it some structural support, so they, for the most part, hold it in place, but it's not rigid. It is a little bit flexible. There's a role of metabolism, so taking our glucose and making ATP. They can form some scar tissue, which is going to come in later when we talk about regeneration of nerve cells. And then they're going to get rid of some excess ions, as the case may be. But one of the big things they do is form that blood-brain barrier, a barrier between our capillaries and our blood and our neurons. And so there's certain things we don't want to let out, we don't want to be able to get out, and certain things we don't want to leave. And so that's one of the primary functions you can see bolded in here. So that's the astrocyte. Next is the oligodendrocyte, kind of looks like an eyeball, and its function is to produce myelin. So we'll raise this picture or enlarge this picture right here. And I'll move it here in just a second so you can see a little bit better. But here you can see a neuron right here. There's an axon of another neuron right here. And the oligodendrocyte is actually right here. Looks like an eyeball. And it has extensions coming off. And those extensions can go towards multiple axons. Remember, the Schwann cell could only wrap around on an axon and produce myelin on one axon. Here you see the oligodendrocyte. It can wrap around multiple places, like this one does here, wraps around this axon and this axon actually in two different places. And so the cell wraps around, and then inside that neurolema, we produce the myelin. Remember, the myelin, as you can see right here, is that lipid material, that fatty sheath that protects the axon and causes the electrical impulse to move much faster. Next, we have microglia cells. Microglia cells are like little spiders. There's one right here. There's one down here. So they're little tiny structures that look like spiders, and they're phagocytic. So they eat up cellular debris. They get rid of bacteria. They get rid of anything that's in our interstitial type cells of our brain. And so they're going to help fight off infection and disease. So these are the microglia cells. Next, we have epidermal cells. Epidermal cells are epithelial type cells that line our brain and spinal cord and produce cerebral spinal fluid. So here we are, cerebral spinal fluid is being produced by these cells right in this area right here. And so these are the epidermal cells. They're an epithelial cell. We find them in our ventricles. There are 4 chambers in our brain, and in the spinal canal, a hollow tube that runs in the middle of our spinal cord that has cerebral spinal fluid flowing through it. Lastly, we're going to talk about regeneration and what happens if a cell gets severed or cut or whatever the case may be. If the injury occurs at the soma over here, the cell's going to die. If the damage occurs in the axon, we can regenerate that. So here we have a small cut. On this end, you can see all of those cells are dying, but what's left behind is sort of like a tube type structure. And then we have our astrocytes produce some growth factor, some neural growth factor, allowing the cell to regenerate and make the connection. Now, sometimes the connection can be reestablished exactly where it was, sometimes not exactly where it is. So the person may see a little bit less control. Maybe it's a skeletal muscle, like in this case, a little loss of control of that tissue. So that's the lecture for chapter 4.